Today we are discussing this important topic of in biochemistry HMP pathway. HMP or hexose monophosphate pathway. It's a very important topic. So let's go. So we start with the other names of this pathway. So first is this hexose monophosphate. Hexose 6 carbon monophosphate, single phosphate. Hexose monophosphate pathway. Okay, so the other names we need to know here. It's also called pentose phosphate pathway. Pentose phosphate pathway, hexose monophosphate or pentose phosphate pathway is also called Dicken-Horecker pathway. Dicken-Horecker pathway. Okay, also called phosphogluconate pathway. Phosphogluconate pathway. Okay, these are the other names here. The site of this HMP pathway is our cytoplasm. So remember, this pathway do not produce any ATP. Do not produce any ATP. Instead, it produces carbon dioxide. CO2 is produced. Do not produce any ATP. Instead, carbon dioxide is produced. So before going to the steps of this pathway, just know the phases of this pathway. Okay, there are two phases of this pathway. The first phase is the oxidative phase. Oxidative phase, which is irreversible phase. Irreversible. And the next step is non-oxidative phase. Non-oxidative phase, which is reversible. Non-oxidative phase here is reversible. Oxidative phase, first phase irreversible. Non-oxidative phase reversible. So let's start with the steps here. Okay. First we will go with oxidative state. Oxidative phase. Oxidative phase. Okay. Yeah. This is irreversible. Okay. And this phase takes place in, remember, liver, adipose tissue, gonads, and adrenal glands. Liver, adipose tissue, gonads, and adrenal glands. So let's start with the steps. First is we have here glucose 6-phosphate. Now this is converted to 6-phosphogluconate. Okay, we'll first write the steps, then we'll write the enzymes. Then 6-phosphogluconate is converted to ribulose 5-phosphate. Ribulose 5-phosphate. Okay. Now from glucose 6-phosphate to phosphogluconate, the enzyme here is G6-phosphate, glucose 6-phosphate dehydrogenase. Here a single NADP plus molecule is used to produce NADPH. Very important first part, the first reaction. Then from 6-phosphogluconate to ribulose phosphate, 6-phosphogluconate dehydrogenase. 6-phosphogluconate dehydrogenase. And here, you know, this ribulose 5-phosphate is a 5 carbon and this is 6 carbon. 5 carbon. So here a carbon dioxide is given off. Carbon dioxide given us, and this carbon dioxide is produced in the reaction. Okay, 6-phosphogluconate dehydrogenase, a carbon dioxide given off to produce ribulose 5-phosphate. Again here NADP plus is used to produce NADPH. Okay, so from these two reactions, this was the whole step of the oxidative phase, which is irreversible. From this reaction we have seen that this phase this phase generates NADPH. Very important point. This phase, and in general the hexose monophosphate pathway generates NADPH. So why we are discussing giving it so much importance? So we'll see the biochemical significance. Biochemical significance of NADPH. Okay. First point, it is required for free radical scavenging. Free radical scavenging. Okay, in RBC and in lens of the eye. Lens of eye. This NADPH is required for free radical scavenging. Free radical are dangerous species causing injuries. Okay, then it is also required for keeping iron in reduced state. What do you mean by reduced state? In hemoglobin in the Fe2 plus state. Fe2 plus state is the reduced state in the hemoglobin, as to prevent methemoglobinemia. Methemoglobinemia is prevented by NADPH by keeping the iron in the reduced state in the hemoglobin, that is in Fe2 plus state. The final significance here is reductive biosynthesis of fatty acids. In biosynthesis of fatty acids, we will see how NADPH is required, and also steroid hormones. The first phase we have seen here, this takes place in gonads and adrenal glands. Mainly here the steroid hormones are produced. Testosterone, estrogen and progesterone in females. In adrenal glands, the steroid hormones. So there is reductive biosynthesis of fatty acids and also steroid hormones. Prevents methemoglobinemia, free radical scavenging. Very important points. Okay, biochemical significance of NADPH. So we have seen the oxidative irreversible phase of HMP pathway. Now we will move on to a complicated non-oxidative pathway. Non-oxidative pathway. Okay, this is our reversible pathway. Here reduces pentoses. 5-carbon pentoses are produced. Why I am saying pentoses are produced? Because in the end of the oxidative phase, ribulose 5-phosphate, 5-carbon is produced, and so the ribulose 5-phosphate will act as the first substrate here, will lead to production of different 5-carbon substances, that is our pentoses. It's a reversible path. So it's a bit complicated one, bit a lengthy one, but not difficult, so just keep with me here. Okay, so we know the first substrate is our ribulose 5-phosphate. So we will take 5-ribulose 5-phosphate. Okay, listen carefully. Now upon these 3 ribulose 5-phosphates, different enzymes act in the non-oxidative phase. First acts with the epimerase enzyme to produce xylulose 5-phosphate. Okay, an action of epimerase, xylulose 5-phosphate is produced. On the second ribulose 5-phosphate, ketoisomerase acts. Ketoisomerase to produce ribose 5-phosphate. Okay, after xylulose 5-phosphate, ribose 5-phosphate is produced. And the third ribulose 5-phosphate, again epimerase acts. Okay, again epimers acts to produce our again xylulose 5-phosphate. Okay, not to be confused, just keep on going with me. Okay, now these are 5 carbon, obviously this is 5 carbon and again 5 carbon. Now the main mechanism will go on between xylulose 5-phosphate and ribose 5-phosphate. So let's go with this. Now there occurs this type of reaction. They combine together and then separate together to form different two products. Okay, what is the enzyme acting here? The enzyme acting here is transketolase. Transketolase. There is transfer of products between these two, so there is trans and ketolase enzyme. Okay, this transketolase enzyme requires magnesium and thymine pyrophosphate TPP or active form of vitamin B1 requires magnesium and thymine pyrophosphate, which will produce eventually glyceraldehyde 3-phosphate, and we know this is a 3-carbon compound. Okay, we know these two 5-carbon compounds are combined here in the presence of transketolase enzyme. So total here is 10-carbon compound. Okay, just to show here, don't write 10-carbon compound, this is a total 10-carbon compound, but here only 3-carbon compound is produced, so remaining 7-carbon compound is remaining. So the 7-carbon compound is pseudoheptulose 7-phosphate, pseudoheptulose 7-phosphate, which is a 7-carbon compound. 3-carbon, 7-carbon, 10-carbon compound is formed. Glyceraldehyde 3-phosphate 3-carbon, pseudoheptulose 7-carbon. Again, following this step, these two compounds combine to produce two different compounds. And what is the enzyme here? Previously it was transketolase, here it is transaldolase. Okay, transaldolase, nothing peculiar about this enzyme, just remember the enzyme is transaldolase. And what are the products? First is fructose 6-phosphate, and we very commonly know 6-carbon, and there is remaining 4-carbon of the total 10-carbon here. So this will be erythrose 4-phosphate, which is our easy 4-carbon. Okay, so this was the total reaction upon here. Following this reaction, the action of transketolase and then transaldolase, this fructose 6-phosphate will be converted to glucose 6-phosphate. Okay, this fructose 6-phosphate work is over. Now we have seen that this action of epimerase, this xylulose 5-phosphate 5-carbon is remaining. Now this will enter the reaction. Xylulose 5-phosphate 5-carbon. This will enter the reaction with erythrose 4-phosphate. Okay, very simple, nothing confusing here, to produce two different compounds. And the enzyme again here is, remember, transketolase, and we know requires what? Magnesium and vitamin B1, which is our thymine pyrophosphate. Okay, now we see this is here 5-carbon, this is here 4-carbon, so total 9-carbon compounds will be produced. Okay, and how we'll differentiate it? A 3-carbon compound will first produced, that is our glyceraldehyde 3-phosphate. Three carbon is gone, remaining 6-carbon. What will be produced? Fructose 6-phosphate. Is of 6-carbon. Okay. Now glyceraldehyde, this glucose 6-phosphate will finally enter the pathway. Okay. Glyceraldehyde 3-phosphate is 3-carbon, which is equal to half of 1 glucose 6-phosphate. half of glucose 6 phosphate is a 3 carbon compound. Okay, and this fructose 6 phosphate is again converted to finally glucose 6 phosphate. Finally, altogether 3 glucose phosphates and their different forms are produced. So this was total non-oxidative reversible pathway of the HMB pathway. Seems a lot complicated, but upon revision to 5 times maximum with writing practice, this will be very simple. The 3 key enzymes should be utmostly remembered here. Transaldoles first, transketolase first, requiring magnesium and TPP, transaldolase in the second, and transketolase again in the third. And the first reaction to epimerase and ketoisomerase. Okay, nothing too complicated here. If you write properly, you will understand everything. This was the total of the HMP pathway. Okay, what is the biochemical significance of this non-oxidative phase? Produces pentoses. Pentoses are required in uric acid cycle. Okay, produces pentoses, and where the organs where it takes place? Organs with rapid cell turnovers, like or liver, not liver, bone marrow, okay, skin rapidly get replaced, and intestine. In these 3 areas, especially this non-oxidative phase takes place with organs with rapid cell turnovers. Okay, now we'll see some clinical significances here. Clinical significances. First is the first enzyme we have seen above in the oxidative phase, converting glucose 6 phosphate to phosphogluconate. This enzyme, glucose 6 phosphate dehydrogenase. This is commonly called G6PD, glucose 6 phosphate dehydrogenase. We will discuss about its deficiency first. Okay, the clinical significance. First is glucose 6 phosphate dehydrogenase deficiency, G6PD deficiency. Very important topic discussion. See carefully. This is the most common enzyme deficiency in humans. What is the second most common enzyme deficiency? We have read in glycolysis. Yes, pyruvate kinase deficiency. Second most common deficiency, pyruvate kinase deficiency. The most common number 1 is G6PD. Okay, it's an X-linked recessive disorder. It's an X-linked recessive disorder. So usually only males get affected. Only males get affected in this X-linked recessive disorder. Only commonly males get affected. Okay. Now there is this fava beans. Okay, there is ingestion of fava beans. Ingestion of fava beans. This ingestion of fava beans aggravate the condition. This phenomenon is called favism. Excuse me, I will write this one again. Favism. Ingestion of fava beans will aggravate the condition of G6PD deficiency, called favism. Also, drugs like sulfa drugs like primaquine. So these two factors aggravate the condition of G6PD deficiency. Sulfa drugs like primaquine and fava beans ingestion. This deficiency is prevalent in Mediterranean, okay, and Middle East region. So the genetic composition of those people is prevalent in Mediterranean and Middle East region. So these regions have high prevalence of Plasmodium. Mosquitoes bite a lot of people, have high prevalence of Plasmodium in this region. This falciparum malaria. Falciparum malaria. Plasmodium causing this type of malaria. So this region have high prevalence of Plasmodium. Okay, prevalent in Mediterranean and middle region. This G6PD deficiency. Now the clinical features here. Okay, what is the clinical feature in G6PD deficiency? So there is decreased generation of NADPH, the single most important compound produced in the oxidative phase of HMP. Decreased generation of NADPH is a free radical scavenger, we have discussed before. Radical scavenger. This will affect RBC membrane integrity. It will affect RBC membrane integrity, which leads to hemolytic anemia. And following hemolytic jaundice. RBC's membrane integrity will be lost, RBC's rupture leads to hemolytic anemia and hemolytic jaundice. Also, this NADPH helps in keeping the iron in its reduced state, that is Fe2 plus state, and due to decreased generation of NADPH, this Fe2 plus converted to Fe3 plus state, causing, it was preventing methemoglobinemia, here it's causing methemoglobinemia. So methemoglobinemia and hemolytic anemia and hemolytic jaundice are the two important clinical features of G6PD deficiency. G6PD deficiency, it is the most common enzyme deficiency in humans, which is followed by our pyruvate kinase deficiency, pyruvate kinase enzyme of glycolysis. So this was all about our discussion on HMP pathway, oxidative phase and non-oxidative phase. Generation of NADPH, very important. Then generation of pentoses required for uric acid cycle. And G6PD deficiency, its clinical features, all have been discussed here. Very important topic. Keep revising them, guys. Thanks for watching. I will catch you in the next one. Good luck.